I'm going to show you an example here how to use Plex to map pathways to cell lines and tumor types, potentially for patient selection and patient stratification. And you'll see it's very flexible in terms of going back and forth between gene expression profiles, pathways, tumor types, cell lines, et cetera. And for this example, we're going to start off using these hallmark pathways. So we have one based on the androgen pathway, and I can select this, and I can run a search based on a pathway as the input. And let's go ahead and do that. In this case, I'm actually just going to export the genes. So you can export this as CSV or Excel. I did this as Excel, and I'm going to go copy those genes that I've exported, and I'm going to paste this back into Plex as an expression profile. So we're going to basically use this list of genes, these 100 genes here, which are androgen responsive, and we're going to use that as our input for the search. And so the question here is, what gene signatures is this signature similar to? So are there other gene profiles that resemble an androgen response? And if so, what are those? We've integrated here all the major gene expression databases, so MSigDB, Geo, Expression Atlas. We also have the entire cancer cell line encyclopedia. So it's RNA-seq profiles, so RNA, protein, proteomic profiles, copy number modulation, essential genes, mutational data. The essential genes are the gene dependency that, like you'll see at the depth map, it's basically the depth map data. And so what we have here, so I put in the genes, and what we can do is look at things like expression atlas, expression profiles, and see where have we seen a gene signature like this before. And what you can see is that we actually see this in other places like MSigDB and Geo, that there's other expression profiles that resemble this androgen response, and in fact, those are also profiles of other androgen responses. So the first one is androgen, the second is androgen. You can see pretty much all these top results. The fourth and the fifth are all some kind of androgen response. And for any of these, you can click on them and see what's actually behind it. So what are the genes that are in the overlap between our signature and this other independent androgen response? In this case, out of 100 genes, 69 of them are also modulated in this other androgen response here. So we can see which genes those are. We can get back to the original source, and that's the case for all of these. So this is a way to identify sort of the identity of the signature. What biology are we capturing? Also a way to identify potential modulators. You can see here dihydrotestosterone, R1A81. So it's a way to identify potential modulators of the signature. And of course, if you know the targeted mechanisms of those modulators, as we do in these cases, it also tells you potentially targets and mechanisms that may modulate your input signature. Okay, and we can go from here over to cell lines, and we can see, are there cell lines, for example, that upregulate this signature? And you can see that there are, in fact, so VCAP cells and LINCAP cells, which are both prostate cancer cell lines, seem to upregulate the signature at both the protein and the RNA level. And you can click on these and see which genes those are that are both in my signature and also upregulated in this VCAP cell line. And you can see 11 of them are upregulated here I think this is at the protein level. So yeah, it's at the protein level. So we can see that prostate cancer cell lines are cell lines that are modulating the androgen response pathway, and therefore could be, prostate cancer could be a tumor type to target if you have something that's modulating the androgen pathway. What you can do here further, which is really pretty interesting, is we've shown inputting pathways, genes, anything that's a result in a Plex search can itself be a search. So in this case, we can actually run a search with cell lines, and I'm going to run a search then with VCAP and LINCAP upregulated genes. So let's go put that into a new tab here. So I can type in VCAP as a cell line. You see all the different features here, essential genes. We're going to look for upregulated proteins, and let's do the same thing for upregulated RNAs here. And let's do this for LINCAP cells. So we can do upregulated RNAs, and let's do upregulated proteins as well here. And so now we've got the search for VCAP and LINCAP cell lines, genes that are upregulated in those cell lines at either the RNA or the protein level. And I'm going to go over and make sure that we can find related cell lines and other things. So we can run that search, and then we're going to see So it's taking these cell lines now and these particular genes that are upregulated. And you can see, first of all, on the target page, at the top of the list will be the genes that are most commonly upregulated in these four sort of, well, the two cell lines, either RNA or protein. And the top result is androgen receptor. You can see that this is upregulated at both the protein and the RNA level in both of these cell lines, which is why it's pushed up to the top. And then we can see things like, what diseases might this be relevant for? You see androgen sensitivity syndrome, prostate cancer, et cetera. So a number of clearly relevant diseases. We can look at things like expression profiles. Actually, let's come back to that. Cell lines, where, what other cell lines have a similar set of upregulated genes? And you see here we've got other prostate cancer cells, and as well as breast cancer cell lines. And if I click on these, it tells me, which genes are in common that are both upregulated in the input cell lines, as well as this cell line. And then we have, if we come over to expression profiles, we can ask where this is, what other expression profiles are these particular genes linked to. And you can see here, there's some potentially interesting links to other instances of prostate cancer's gene expression profiles. In this case, around modulation with this BET bromodomain inhibitor, and others as well, as you see going down. And again, you can click on these and see which are the genes that are upregulated in my cell lines, which are also modulated in this other independent expression profile. So hopefully I've shown you how you can really flexibly go from a pathway to genes that are modulated to places we've seen that same gene signature before, whether it's expression profiles, whether it's cell line profiles, and how we can use that kind of data to map to particular tumor types, cell types, and particular patients potentially based on the cell type or based on their gene expression profile. If you're interested in learning more, please reach out, maybe check out our website at plexresearch.com. Be happy to talk to you in more detail about how this kind of approach may be useful for your oncology projects. Thank you for your time.